Okay, this lecture is on transport in plants, which is our topic of our next lab, transpiration. So plants get the water and minerals that they need from the soil. They need water to do photosynthesis, they need water to transport materials, they also use it for evaporative cooling, and they also use it for internal pressure, and which gives them support and makes it so they're turgid and not wilting. The problem plants have is that they lose a large amount of water due to evaporation, and all of that water must be replaced. So they need to constantly be bringing water up from the soil throughout the plant. And if you think about some of the tallest trees, that's quite a bit of a distance. And if you think about all those leaves that they have, they're losing lots and lots and lots of water every hour on a hot, sunny, dry day. So they have to constantly be moving this water. So we already talked about this, so I'm not going to spend much time on this. There was another lecture on water potential, but remember that water potential is the sum of the solute potential, which has a negative effect on water potential, and the pressure potential, which has a positive effect on water potential. And remember that water potential is the potential of water to leave a particular area. And so this just shows you water potential in a plant, and so the plant on the left has negative water potential, and because it has a high negative solute potential, and there's no pressure potential. And then on the right, the plant has a water potential, and because it has a negative solute potential that is balanced by a positive pressure potential. And so then you get the plant to be turgid. So there's a pressure of the cell membrane on the cell wall. Because it's filled, the vacuole is filled with water. Okay, so how plants actually move things, materials, there's a couple of ways. One is by bulk flow, and this is actually using that pressure potential I was just talking about. So plants will sometimes move ions to a particular area. Remember, water follows solute, so as water fills that space, it causes pressure. Increased volume causes pressure, and that can actually create the movement of water, so that can increase the water potential by increasing the pressure. And remember, pressure has a positive effect on water potential. So that's one way that we see materials moved. And plants have what are called aquaporins. We were introduced to that in chapter 6. I'm not sure what chapter it was, but this last chapter we just did on cell membranes. But these are just channels. They're proteins embedded in the cell membrane where water can pass through readily. And plants have these not only in their plasma membrane but also in their tonoplast. And the tonoplast is the membrane on the vacuole. And so the plant can actually regulate water movement by changing the number of aquaporins that are present. Remember, water movement can be regulated, so we can do things to increase or decrease the rate that the water is moved, but you can't change the direction. Water is always going to move passively, so water is always going to go from high potential to low potential. And remember, too, that minerals, ions, cannot pass through membranes without transport proteins, and the ions that are in the soil are generally at a lower concentration than in the plant. And so they must be taken up by active transport. So the plant has to expend energy in order to move those ions from the soil into the plant. And so the plant uses a combination of gradients in order to make this happen. So it can use electrical gradients, and it can use concentration gradients. And together, we're going to see these are often coupled. We call this an electrochemical gradient. And if you're going against the concentration gradient, then you need to utilize ATP. So in animals, what we see for the most part for these pumps are sodium-potassium pumps. And a lot of the ATP, most of the ATP that you generate actually goes to run these sodium-potassium pumps. In plants, however, what we see are proton pumps. And so what the plant does is it expends energy to move protons out of a cell against the concentration gradient. So here we have, if I can get my little pen. So here we have a proton pump embedded in the cell membrane. This is just a protein, and so we're going to expend energy. So the plant is utilizing ATP, and it's pumping protons against the concentration gradient. So now we have more protons out of the cell than inside of the cell. we also have created an electrical gradient because protons have a positive charge. And so now, relatively speaking, the outside of the cell is going to be more positive and the inside of the cell would be more negative. So as a result, potassium ions will go down through a potassium channel, and this is with the concentration gradient or the electrical gradient. So it's actually a form of facilitated diffusion. There's no energy expended in order to do this. So now these potassiums flow down, and now we have a symport or a protein that's going to pump two things at the same time together. And so now as the the protons go down their concentration gradient, they carry with them the negative chloride ions. And so that's the way that the plant can move these chloride ions into the cell. The inside of the cell had more chloride ions than the outside of the cell. All right. Okay, so there was a lot of research on how water gets moved in a plant. Is it being pushed or is it being pulled? And there's actually evidence of both. Guttation is from root pressure. So here is just water is forced through the little pores in the leaves, and that is just due to the pressure in the roots. So generally that's something where you're going to have maybe a change in the humidity or a change in atmospheric pressure, and that's going to allow that atmosphere. The other is a pulling, and this is the majority of the way that materials are moved through a plant, and that's through transpiration, and that's what your lab is about. And so what happens here is you get water loss at the leaf, and that creates a pull behind it, and that pull just kind of gets followed throughout the plant. So if we take a cross-section of a leaf here, and this would be the surface of the leaf and has a waxy cuticle on it, so this is where the sun would be hitting it. That's followed by this mesophyll layer, and this section here, I don't think you need to know the names of them anymore. And then this area, and this is where most of the photosynthesis is occurring. And then down here, there's a lot of air spaces in the leaf. And this is also where you're going to find the veins of the leaf, and the veins are going to contain those cells that are involved in transport. So they're going to have the cells that are going to be lined up to bring water up from the roots. And then at the bottom of this type of leaf, we find these stomates, and I have some slides later on that I'll show you on those. But they're basically just pores in the leaf where water and gases can be exchanged. So here carbon dioxide can enter for photosynthesis, oxygen gas can exit, but because there's this pore, water will also leave from this area just due to evaporative loss. So what ends up happening is we end up with this like negative pressure. So if the water a water molecule is lost at the leaf here at this pore, then behind that the water film that's along the cell walls of the mesophyll, it ends up getting a greater curvature, right? So if we lose some of the water, we're changing the curvature of this film of water. And that creates negative pressure. And then if we lose a little bit more water, we get even more negative pressure. And what that does is it pulls water in from the surrounding cells. And then eventually from the cells that are transporting the water up to the leaf, and each cell, each kind of behind that, and it just keeps pulling to causing this negative pressure. And that's how we get water flowing from the roots all the way up to the leaves. And so this is just another picture kind of showing that. So we have water coming up from the ground and it's being evaporated at the leaf. And so we get this a little bit of evaporation occurring here. And again, that's going to cause this pulling from the cells in the mesophyll as the water is lost from the cell wall. And that's a mesophyll cell. And here's our vein, which is transporting the water. And so you lose this water through just evaporation. And then the water evaporates from the mesophyll cell walls to replace that, and then eventually that's going to pull from the veins into the apoplast of the mesophyll cells, into the cell wall, the membrane, into the cell wall. And then behind that it's going to pull the xylem, the water that's in the actual xylem or the veins of the leaves, and that's going to continue all the way down to from the stem and upward and from the root. And eventually it'll be pulling the water from the ground. Here's our xylem, which is transporting our water, and the water that's surrounding the root. And that's and then we have the property of cohesion, where the water molecules are attracted to each other. So these tubes are very, very, very thin, and that's going to help to pull the water up from the ground as well. And here we just have our little water molecules attracted to each other. Okay, and so this whole process is a passive process. The plant does not have to expend any energy to move the water from the ground. It's all osmosis. And the water enters the root from the soil by osmosis. Okay, the other thing that transpiration does, although it's not its primary function, it also does help to cool plants. So as water evaporates, just like water evaporating from us has a cooling property, it does the same thing for the plant. And so that can keep the plant at lower temperatures when in very hot environments. Okay, these pores that I was talking about where the water is being lost are actually called stomates, or a stomate is just a hole, an opening in the leaf. And it's surrounded by two cells which we call guard cells. And these two guard cells control the opening and closing of this stomate. So they ultimately control how much water is evaporating from a particular plant at a particular time. And so here we have a picture of the stomate. And so what the plant does is it pumps the protons out of the stomate actively. So it's going to expend energy to do that. And remember I showed you before, then potassium ions follow that. And then when the hydrogen ions go down their concentration gradient, they take with them the chloride ions. And then remember that water always follows solute, so as we increase the concentration of the ions inside the stomate, water follows. And as the water flows into the stomate, the cells swell. And since they're kind of attached to each other on either end, when they swell, it's like having two balloons. And if you increase it, you're going to increase the opening in between the two of them. So this is one cell, and this is another cell, and as it swells, it opens up the stomate. And then as the ions leave, water leaves, and then the stomate closes. That's for that. And this is a picture, an electromicrograph of the stomates. So here are just some mesophyll cells, and this is a guard cell, and this is another guard cell, and this little opening right here is the stomate. And I think they look a little bit like eyeballs when you're looking at them under the microscope. So plants can control when their stomates are open or closed. The problem is they need the stomates open in order to get carbon dioxide in order to do photosynthesis, but it's a balancing act. So at night they close their stomates. Since there's no sun and they're not doing photosynthesis, they don't need carbon dioxide, so they can control that there's no water loss. And then during the day, the stomates will open to do photosynthesis. But the plants are regulating how much water is being lost, and if the water is being lost too rapidly at the hottest time of the day, then the stomates may close. So the plants take, not being able to do photosynthesis, but it's maintaining that balance of water. And you're going to be in the lab manipulating something that might control the speed of which transpiration occurs, or whether the plant is opening or closing stomates. And so they can control the water loss by a couple of ways. One is by opening and closing their stomates, and the other is by controlling how many stomata they actually have. And actually we see where the stomates are found as well. So certain plants, depending on their environment, may have more stomates. They may have stomates on the surface, the top of the leaf. They may have the stomates on the bottom of the leaf. And all of this is just to maintain